In Activity 9, Population Experiments, students try to determine exactly what causes a daphnia population to change. They first examine their team's daphnia population graphs and discuss how the populations are changing, and then observe the results of their experiments and draw conclusions. Finally, students graph the results of their experiments. You will need the following materials from the kit: Activity Sheet 8, Activity Sheet 9, daphnia counters, droppers, magnifiers, vials with caps, container of green algae, baster, daphnia population data graph chart, 10-ounce containers, container of daphnia culture, extra daphnia water, and masking tape. You will also need to provide felt tip markers and spring water. Begin this activity when the team's daphnia populations begin to decline. To prepare for the activity, make 2 copies of Activity Sheet 8 and 1 copy of Activity Sheet 9 for each student. Pour 2 to 3 centimeters of daphnia water into each 10-ounce container. Then, use the baster to transfer 10 daphnia to each of these containers. Each team of 4 will need 2 vials with caps, a 10-ounce container with 10 daphnia in water, 2 pieces of masking tape, a marker, 2 droppers, and 2 magnifiers. To begin Session 1, ask students, what causes a population to increase? Students should recall that a population increases when the individuals reproduce. Explain that at the same time that some individuals are reproducing, other individuals are dying. Point out the varying height of the bars on the class chart, the daphnia population graph, and ask students, how has the class's daphnia population changed? The students should respond that the classroom population of daphnia has decreased. Then ask, what do you think has caused the population of daphnia to decrease? Students should reason that more daphnia have died than have been born. Finally, ask students, what are some of the possible reasons you can think of that the daphnia have died? Students are likely to mention lack of food, too much or too little light, water that was too hot or too cold, and lack of enough space. Next, ask students, how could you find out more about what conditions are ideal for the daphnia to live in? Students may suggest trying different experiments to see what happens under different conditions. Explain that a variable is a condition that can be changed or varied in an experiment. A good example for students would be to set up an experiment in which they would give each of their daphnia a larger space to move around in. Ask students, in this experiment, what would be the variable? The amount of available space would be the variable being tested. Then explain that the control is the part of the experiment in which everything is kept the same, and the experimental setup is the part in which one variable is changed to test it. Ask students, what would be the control in your experimental setup to test the effect of available space on a daphnia population? Guide them to understand that the control would be keeping the daphnia in the vial in which they are living now, with nothing changed. Then ask students, what would you change in your experimental setup? Would you simply put them in a larger container? Some students might say yes, but others will realize that they would not really have more living space if they were in the same amount of water as before. Ask students, what if you only added more water to the vial in the experimental setup? Would this give each daphnia more living space? Students should realize that it would. Remind students again that only one variable should change in each experimental setup. Everything else should be kept the same as in the control. Next, go down the list of possible reasons for the decrease in the daphnia population, and for each one ask, how could we test this condition to see how it is affecting the population of the daphnia? In each instance, remind them that it is important to set up 2 parts, a control and an experimental setup. Then divide the class into teams of 4 and allow each team to choose a variable to test. Be sure that at least one team is testing each of these variables: temperature, amount of light, amount of space, kind of water, and amount of food. Next, give a copy of Activity Sheet 9 to each student and distribute the materials. Have teams label their vials with their team number, today's date, and either the letter C for control or E for experimental, and set up their experiments. Inform students that each vial should contain 5 daphnia. Instruct each team to describe and draw their experiment and predict its results on their activity sheets. Have them begin filling in the chart on the activity sheet with the observation day number 0, today's date, and the present population 5 in each vial of daphnia. Finally, collect the materials and return them in the kit. Collect the activity sheets for use in continuing observation sessions. To conclude Session 1, every second or third day, have students count the daphnia population in both their control and experimental setups and record this information on their activity sheets. To begin Session 2, return their copies of Activity Sheet 9 to students. Distribute 2 droppers, 2 magnifiers, 2 daphnia counters, and 2 10-ounce containers to each team of 4. Tell students to complete the activity sheet with the results of their team's experiment. Then, distribute 2 copies of Activity Sheet 8 to each student and have them make 1 bar graph of their control and another of their experimental setup, using the numbers on their chart on Activity Sheet 9. Finally, discuss the results of their experiments. Have each team show their bar graphs to the class and describe what they did and what the results were. Encourage other students to ask questions of the team reporting. If class results differ from the expected, discuss the possible reasons for the difference. It is more likely that they have used a faulty experimental design than that they have a strain of daphnia that is not reacting typically. If necessary, suggest they set up their experiments again as carefully as possible and repeat them. To conclude Session 2, return the daphnia to the main container. Collect the materials, rinse and dry them, and return them to the kit. For science background, reinforcement activities, curriculum connections, and information about the Delta Science Reader, please consult your DSM Teacher's Guide.